TSIA, triple sugar iron agar. The TSI test is used to determine different fermentative properties of bacteria as well as the ability to produce hydrogen sulfide. TSI agar contains 3 sugars: lactose, sucrose, and glucose. It also contains thiosulfate as a sulfur source for the thiosulfate reductase enzyme produced by some bacteria. Glucose is present in TSI in very low concentrations. Glucose is a monosaccharide. Lactose and sucrose, both disaccharides, are present in the medium in very high concentrations. This will allow differentiation between monosaccharide and disaccharide fermentation. Hydrogen sulfide production will be observed by the presence of a black precipitate in the bottom of the tube. When inoculating the TSI, you use a stab streak method. In this method, the thick bottom part of the tube, called the butt, will actually be punctured with the needle. The needle, when removing it from the butt of the tube, will then be run slightly across the slant. Using a needle will help avoid over-inoculation. Notice here that the needle actually punctures the agar, then comes up the slant afterwards. This technique introduces anaerobic and aerobic conditions as well as prevents over-inoculation. Here are some of the more common results seen with the TSI test. The tube at the top in this picture is a control. On the bottom is a disaccharide fermenting bacteria in the TSI. Obviously, there is some gas production. Phenol red is the pH indicator used in the TSI test, so fermentation will produce acid products, causing phenol red to turn yellow. Here, the bottom tube is alkaline. This is a non-fermenting bacteria. Protein catabolism gives off alkaline conditions. Alkaline conditions will cause the phenol red to turn a bright pink. Now shown is a property of some bacteria known as reversion. Reversion occurs with glucose-only fermenting bacteria. Since glucose is present in very low concentrations, it quickly runs out, causing the bacteria to revert to other forms of metabolism, such as protein catabolism. This will result in the bottom of the tube turning yellow from acid, byproduct of fermentation, and the slant turning a bright pink because of protein catabolism that is occurring. The top tube is a control. Now shown is hydrogen sulfide production. Hydrogen sulfide gas is produced by some bacteria that use the enzyme thiosulfate reductase. Thiosulfate reductase will reduce the thiosulfate in the media producing hydrogen sulfide gas. The hydrogen sulfide gas will then react with the sulfur indicator known as ferrous sulfate, or an iron salt. This causes a black precipitate to form. It is important to note that hydrogen sulfide production will only occur in acidic conditions. This means that fermentation of some form must have occurred in order for hydrogen sulfide to be produced. Both of the organisms shown here are hydrogen sulfide producers, or H2S positive. The organism on the top is a glucose-only fermenter. The yellow from fermentation, shown by phenol red, is disguised by the black precipitate from sulfur reduction. In this case, the tube at the top is an example of reversion. However, it is H2S positive instead of negative. The tube on the bottom is acid-acid. Both the slant and the butt are acidic, and it is H2S positive. Remember, if the organism is H2S positive, acidic conditions must have been present at some point in time in the tube in order for the thiosulfate to be reduced. Now shown is the alkaline acid H2S positive organism with a control. During incubation, condensation can often build up in the bottom of the slant. Here, even though a small amount of black precipitate is shown, this is not considered positive for H2S production. In this case, the ferrous sulfate has reacted with some water that has collected at the bottom of the slant, causing the black precipitate to form. In correct technique, a needle is used to avoid over-inoculation of the TSI slant. Using too much bacteria can cause all three sugars to be quickly used up in the very top of the slant. This can sometimes be confused for reversion or alkaline acid conditions when in fact all three sugars have been used. Avoid over-inoculation by not using a loop, and when running the needle up the slant, remove it from the slant right before you reach the very top. This will prevent misinterpretation of your test results. The TSI is very helpful in differentiating monosaccharide versus disaccharide fermentation. It is commonly used for the identification of gram-negative enteric bacteria, although it can be used for many gram-positives. The added result of hydrogen sulfide production is also helpful in differentiating between different species of bacteria. The TSI can also be used to confirm or in conjunction with phenol red broths.